I'm Kathy. I'm a biomedical technology student, and I have quite an inquisitive mind. I hope you do too. Now, have you ever wondered what blood types you actually are? And if you do, have you ever wondered what actually goes on in the lab to determine the blood type that you actually are? If I spot it interesting, come through and let me show you how it's actually done in the lab. It's quite interesting, really. Now, this process is called ABO blood typing. Now, in order to understand it clearly, you have to understand that amongst other components, blood has red blood cells, which are used to recognize the type of antigens present in your blood. Now, this is what we imagine your red blood cell would look like. Now, you have to keep in mind that the type of antigen present reflects the blood type that you actually are. Now, this is used in forward typing rather. Now, this right here is an antibody. Now, viewing the antibody present occurs in reverse typing, used to confirm the results from forward typing. And we imagine your antibody would look somewhat like this. Now, the antibody present, as you remember, I said, reflects the blood type that you actually are. So if you have your antigen A present, you know that you're blood type A. So you have your B antigen, then you're blood type B. If you have both A and B, then you're blood type AB. If you have neither A and B, then chances are you're blood type O. Now, this right here is how we spot the presence of an antigen or an antibody. This is a reflection of agglutination. An antigen is coated all around it by antibodies. Now, this is agglutination rather under the microscope. Now, here I'll show you how it looks like on a test tube. Now, this is how agglutination is reflected on a test tube. Down here on your blood, which has red blood cells only, don't forget, is granules, a number of granules that no matter how many times or how vigorously you shake the test tube, they will not disintegrate, they do not fall apart. You just have the granules there. Now, that is how you spot agglutination and the presence of your antibody or your antigen. Now, enough about the nitty-gritty parts of it. Now, I'm going to show you how we actually do it, how we actually perform the procedure, but on paper, of course, so now bear with me. Now, this right here is a reflection of a centrifuge blood specimen. So we draw blood from you, and it comes to the lab, and we centrifuge it. Now, centrifugation is another fancy word to say the separation of your blood plasma and your red blood cells. So now, why do we do this? We do this so that we take your blood plasma, we put it in a separate test tube, and we take your red blood cells, a portion of your red blood cells as well, and put it in a separate test tube. Now, you will see the importance of this later on. Now, in the second step of the procedure, we add clean saline up to a quarter of the test tube. Now, we use the centrifuge red blood cell blood specimen plus the clean saline solution, and we mix it a bit and it is centrifuged. Now, why is this done? This is done to wash the cells, to wash the blood cells. Why do we wash the blood cells? To remove any traces of blood plasma that could be on that red blood cell specimen right there that was centrifuged before. So we repeat this 3 times. After centrifugation for the first time, we will add another dose of clean solution of clean saline up to here. It is centrifuged again and again and again 3 times because we are meticulous like that. Everything we do, it has to be on point and it has to be exact like that. So after we have washed it 3 times, removing all traces of blood plasma, we create red blood cell specimen. Suspension rather. Now, this is what your red blood cell suspension would look like. In order for us to use it, it would have to be this beautiful bright red that we can use to recognize or be able to spot agglutination easily. So we take this blood suspension, red blood cell suspension, we put it in test tube 1, test tube 2, and test tube 3. Now that is your forward typing. We add 1 drop in each labeled test tube. Once again, on the 3 test tubes I told you about earlier, test tube 1, 2, 3, the forward typing labeled A, B, and O, you add 1 drop of suspension there, another and another respectively, and then afterwards you add a reagent. So on your test tube 1 labeled A, you add your anti-A reagent. On your test tube B, your anti-B reagent. On your test tube O, you add your anti-A, anti-AB reagent as well. So, if you leave it to incubate for 5 minutes, now after 5 minutes, you will check for agglutination. Well, in my case, I found agglutination in all three test tubes. Now this means both antigen A and antigen B are present. Now we have already discovered that blood type the presence of antigen A and the presence of antigen B makes you a blood type AB. That means my patient right here is a blood type AB. Now we have to confirm these results because everything we do as biomedical technologists has to be accurate. So we also have to do the antibody search. Now on test tube 4 and 5, we have the labeling small a and small b. This is for the presence of antibody A and antibody B. Now, where do you find your antibodies? As we spoke earlier, we know that we find your antibodies on your plasma. So we take the plasma that we had on our centrifuged blood specimen and we add a drop of that plasma on test tube A, a drop of that plasma on test tube B, small b. Now what we do now, as our reagent, we'll add A blood cells on test tube small a and B blood cells on test tube small b. So now in my case, there was no agglutination present. That means there are no antibodies in this blood specimen. Now this confirms my results. As we found out earlier that I have both antigen A and B present in my blood specimen, and I do not have any antibodies present. So that means my patient is a blood type AB. Now that's how ABO blood typing occurs. Thank you.